Hello everyone, welcome to my channel Capsa. Today's topic is T cell subsets. So, where do T cells originate and mature? T cells originate from hematopoietic stem cells located in the bone marrow, and however, they do not mature in there. Immature precursor cells migrate to the thymus, which is a specialized primary lymphoid organ situated behind the sternum. Inside the thymus, these cells undergo a series of developmental and selection process that ensure only functional self-tolerant T cells which will survive. Because of this maturation site, they are called T lymphocytes or T cells, where T stands for thymus derived. How do T cells recognize antigens? Unlike B cells, which can recognize free floating antigens directly, T cells can only recognize antigens that are presented on the surface of other cells. These antigens must be displayed by a special proteins known as major histocompatibility complex or MHC molecules. There are two main types of MHC molecules. MHC class I, which is present on all nucleated cells and interacts with CD8 cytotoxic T cells. MHC class II, which is expressed on specialized antigen presenting cells such as dendritic cells, macrophages, and B cells, and interacts with CD4 helper T cells. And the T cell receptor or TCR on the T cell surface binds specifically to the antigen MHC complex, allowing precise immune recognition. Now, how are the T cells activated? So, for a T cell to become fully activated, it requires two signals. The first signal is the binding of the T cell receptor or TCR to the specific antigen MHC complex presented by an antigen presenting cell. The second signal is to co-stimulatory interaction, for example, between CD28 on the T cell and B7 on the antigen presenting cell. When both these signals are received, the T cell begins to proliferate, means divide rapidly, and differentiate into effector or memory cells. This dual signal system ensures that T cells respond only when truly necessary, preventing unwanted activation. What are the main subsets of T cell and their functions? T cells are not a single uniform group. They are differentiated into several subsets, each with unique roles in the immune response. The five major subsets include helper T cells, cytotoxic T cells, regulatory T cells, memory T cells, natural killer T cells. So what is the role of helper T cells? Helper T cells are identified by CD4 surface marker, which binds specifically to MHC class II molecules. Their primary role is to coordinate and regulate the immune response by releasing signaling molecules called cytokines. These cytokines activate and guide other immune cells such as B cells, macrophages, and cytotoxic T cells. Now, what are the subtypes of helper T cells and their specific functions? So, helper T cells can further differentiate into several subtypes, including Th1, Th2, and Th17 cells. Th1 cells secrete interferon gamma or IFN gamma, which activates macrophages and cytotoxic T cells to destroy intracellular pathogens such as viruses and certain bacteria. Th2 cells release cytokines like interleukin 4 and interleukin 5, stimulating B cells to produce antibodies. These cells are crucial in fighting extracellular parasites and are also involved in allergic responses. Th17 cells produce interleukin 17 or IL-17, which recruits neutrophils to infection sites and enhancing mucosal immunity. However, excessive Th17 activity can contribute to autoimmune disease also. Now the cytotoxic T cells. How do cytotoxic T cells recognize and eliminate infected cells? We will understand this. Cytotoxic T cells marked by the presence of CD8 recognize antigen presented by MHC Class I molecules, which are found on all nucleated cells. These cells play a direct role in killing virus infected or tumor cells. They use specialized proteins such as perforin, which creates spores in the targeted cell membrane, and granzymes, which enter through these pores and trigger apoptosis or programmed cell death. This process ensures that infected cells are destroyed efficiently without causing excessive tissue damage. Now, what is the function of regulatory T cells and how do they maintain immune balance? Regulatory T cells are usually CD4 and express a critical transcription factor called FOXP3 or FOXP3. Their function is to suppress overactive immune responses and maintain self tolerance, preventing the immune system from attacking the body's own tissues, called autoimmunity. They achieve this by secreting anti inflammatory cytokines such as interleukin 10 and transforming growth factor beta or TGF beta, which inhibit the activity of other immune cells. Now, how do memory cells or memory T cells provide long term immunity? So, memory T cells are long lived cells that remain in the body after an infection has been cleared. They are characterized by surface marker CD45RO. Their main function is to provide immunological memory, allowing the body to respond faster and more effectively if the same pathogen enters again. There are three types of memory T cells. Central memory T cells or TCM circulate through lymphoid organs and have a strong capacity for proliferation when they re-encounter the antigen. Effector memory T cells or TEM reside mainly in the peripheral tissues and can act immediately without needing further activation. Resident memory T cells or TRM permanently stay in specific tissues such as the skin, lungs, and mucosal surface, providing a localized and rapid response against the reinfection. Now, how do natural killer T cells bridge innate and adaptive immunity? So, natural killer T cells are unique because they share properties of both T cells and natural killer cells. They express both a T cell receptor and NK cells markers like NK1.1 or CD161. Unlike typical T cells that recognize peptide antigens on classical MHC molecules, NKT cells recognize lipid antigens presented by a special molecule known as CD1D. When activated, they can rapidly release large amount of cytokines, thus linking the innate immunity system, innate immunity's quick response with the adaptive immune system's specificity. So stay updated, stay curious, keep exploring, keep learning. If you found this helpful, don't forget to like, share with your friends, subscribe for more microbiology insights. Thank you.